Welcome to the Veterinary Physiology online class. Today, we will be diving into Unit 3A, which focuses on the endocrine system. Our speaker for this session is Dr. Anamika Pandey from GNP Sir classes. The endocrine system is a complex network of glands that produce and release hormones to regulate various functions in the body. Let's explore the key components and functions of this system. Endocrine glands secrete hormones directly into the bloodstream, influencing various bodily functions. These glands include the hypothalamus, pituitary, thyroid, and more. Hormones act as messengers, affecting different parts of the body. Here, we see a detailed list of major endocrine glands and the hormones they produce. Each gland has a specific role and produces hormones that regulate various physiological processes. Hormones can be classified based on their chemical nature. This slide shows the classification of amines, including epinephrine and dopamine, which are derived from amino acids and have specific functions in the body. Polypeptides and simple proteins are another class of hormones. These include hormones like thyrotropin-releasing hormone and insulin, which play crucial roles in regulating bodily functions. Glycoproteins and iodinated amino acids are also important hormone classes. Glycoproteins like TSH and FSH are essential for various endocrine functions, while iodinated amino acids like thyroxine are vital for thyroid function. Steroid hormones, such as cortisol and progesterone, are derived from cholesterol and play significant roles in metabolism and reproductive functions. Fatty acid derivatives like prostaglandins are involved in inflammation and other processes. Protein hormones consist of amino acids and are generally termed as polypeptides. They play various roles in the body, including growth and metabolism. When conjugated with carbohydrates, they are termed glycoproteins. Steroid hormones are produced in the gonads and adrenal glands. They are synthesized from cholesterol and play crucial roles in reproductive and metabolic functions. The synthesis involves several steps within the mitochondria and endoplasmic reticulum. The adrenal cortex contains enzymes essential for the production of glucocorticoids and mineralocorticoids. These enzymes facilitate the formation of hormones like pregnenolone and progesterone, which are vital for various bodily functions. The pituitary gland, located at the base of the brain, is divided into anterior and posterior parts. The anterior pituitary secretes hormones like TSH and ACTH, while the posterior pituitary releases oxytocin and vasopressin, which regulate various physiological processes. Growth hormone plays a crucial role in growth and metabolism. It affects bone and muscle growth, protein synthesis, and lipid metabolism. Abnormal levels can lead to conditions like gigantism or dwarfism. Antidiuretic hormone, AD, affects water reabsorption in the kidneys and regulates blood pressure. It is crucial for maintaining fluid balance in the body. AD release is influenced by factors like hydration levels and blood pressure. ADH increases water reabsorption in the kidneys and helps regulate blood pressure. It has a less prominent pressure effect compared to oxytocin. Deficiency in AD can lead to conditions like diabetes insipidus. The thyroid gland, located in the neck, produces hormones like thyroxine, T4, and triiodothyronine, T3. These hormones regulate metabolism, growth, and development. The gland's function is influenced by the availability of iodine. Thyroid hormones, T4 and T3, contain iodine and are crucial for regulating metabolism. They stimulate various physiological processes, including glucose metabolism and heat production. The formation of these hormones involves several steps. Thyroid hormones affect almost every cell in the body, stimulating metabolic activity and enzyme synthesis. They are essential for normal growth, development, and the regulation of the nervous system. Hypothyroidism and hyperthyroidism are conditions caused by imbalances in thyroid hormone levels. Hypothyroidism can lead to symptoms like weight gain and fatigue, while hyperthyroidism can cause weight loss and nervousness. Graves' disease is an autoimmune disorder associated with hyperthyroidism. Hormone secretion follows specific patterns, including ultradian, circadian, and infradian rhythms. These patterns influence various physiological processes and can vary between species. Hormones act on target cells through different mechanisms, including endocrine, paracrine, and autocrine actions. They bind to specific receptors and trigger responses that regulate various bodily functions. Hormone receptors can be located on the cell surface or inside the cell. Cell surface receptors generate second messengers, while intracellular receptors alter gene expression. These mechanisms ensure precise regulation of physiological processes. Hormone secretion is tightly regulated by negative feedback mechanisms. This prevents overactivity of the hormone system by adjusting the activity level of the target tissue rather than the hormone's secretion rate. Feedback regulation can occur at all levels, including gene transcription and translation, and involves the hypothalamo-pituitary adrenal axis. The hypothalamus releases gonadotropin-releasing hormone, GnRH, which stimulates the anterior pituitary to release gonadotropins, FSH and LH. These gonadotropins act on the gonads, which then produce sex steroid hormones. These hormones inhibit the secretion of GnRH and the responsiveness to GnRH, creating a negative feedback loop. Positive feedback occurs in specific instances, such as the secretion of luteinizing hormone, LH, based on the stimulatory effect of estrogen. Estrogen stimulates the ovaries to produce more estrogen and the pituitary gland to produce LH. When LH reaches a certain level, the negative feedback mechanism is triggered. Hormones are transported in the blood either as water-soluble or lipid-soluble forms. Water-soluble hormones dissolve in plasma and diffuse to target cells, while lipid-soluble hormones circulate bound to plasma proteins. Thyroxine, for example, is more than 99% bound to plasma proteins. Hormones are cleared from the blood through various mechanisms, including metabolic destruction by tissues, binding with tissues, and excretion by the liver and kidneys. The rate of clearance is measured by the disappearance of the hormone from plasma. The hypothalamus plays a crucial role in hormone regulation by releasing specific hormones that control the secretion of other hormones from the pituitary gland. GnRH is a decapeptide with a molecular weight of 1183 daltons. It induces the release of both FSH and LH. The C-terminal portion of the molecule is necessary for receptor attachment, and the first three amino acids are crucial for activating LH and FSH release. In use, its half-life is 7 minutes. CRH, TRH, and PIH are other hypothalamic-releasing hormones. CRH is a 41 amino acid peptide, TRH has 3 amino acids, and PIH regulates prolactin in mammals. The production and release of these hormones are influenced by feedback systems and external stimuli. TSH is a glycoprotein hormone with a molecular weight of 30,000 daltons. It affects the thyroid by increasing the activity of thyroid cells, including iodide uptake, thyroxine production, and thyroglobulin proteolysis. Low temperatures can increase TSH output. TSH consists of a beta chain of 112 amino acids and an alpha chain of 89 amino acids, identical to FSH and LH. TSH secretion is stimulated by TRH from the hypothalamus and inhibited by somatostatin. It binds to receptors on thyroid cells to stimulate thyroxine secretion. TSH stimulates the thyroid gland to secrete thyroxine, T4, by binding to GPCRs on thyroid cells. Some individuals develop antibodies against TSH receptors, leading to conditions like hyperthyroidism or Graves' disease. ACTH is a polypeptide with a half-life of 6 minutes. It stimulates the adrenal cortex to secrete cortisol and corticosterone. In mammals, it increases glucocorticoid production, while in birds, it is necessary for aldosterone output. Prolactin is a polypeptide with 198 amino acids and a half-life of 15 minutes. It regulates lactation, stimulates pigeon crop gland, induces maternal behavior, and acts as a metabolic hormone. High levels in women suppress menstruation. Gonadotropins include FSH and LH, which have alpha and beta subunits. The alpha subunit is identical for FSH, LH, and TSH. FSH has a half-life of 2.5 hours and is crucial for reproductive functions. LH has alpha and beta subunits with a molecular weight of 30,000 daltons and a half-life of 30 minutes. It induces estrogen secretion and is responsible for the preovulatory surge that leads to ovulation. It also stimulates interstitial cells in both males and females. The preovulatory surge of LH, initiated by estradiol, is crucial for ovulation. This surge is controlled by the preoptic and anterior hypothalamic nuclei. Placental lactogens are molecular relatives of prolactin and growth hormone. They have been identified in primates, ruminants, and rodents, but not in other species. The posterior pituitary stores hormones synthesized in the supraoptic and paraventricular nuclei. Two chemically similar polypeptide hormones are produced here, with 6 identical amino acids and a molecular weight of 1000 daltons. The neurohypophysis produces various peptides, including lysine vasopressin, arginine vasopressin, oxytocin, and vasotocin. These peptides are co-synthesized with N-neurophysin and have specific functions in different animal groups. Oxytocin acts on the smooth muscles of the mammary gland and uterus, increasing contraction frequency. It also induces oviposition in birds and has a milk ejection reflex. Exogenous oxytocin has a luteolytic action in cows and goats. ADH, also known as vasopressin, plays a crucial role in the water reabsorption mechanism of the kidney. It varies among species, with cattle, humans, and most mammals having arginine, while swine have lysine. The half-life of ADH is just a few minutes. The output of ADH is influenced by the body's hydration level, with osmoreceptors in the hypothalamus and paraventricular nuclei playing a key role. Concentrated body fluids trigger ADH release, while diluted fluids inhibit it. ADH increases water reabsorption from the distal convoluted tubule and collecting ducts by enlarging the size of water pores. At normal physiological levels, ADH does not affect electrolyte absorption. A deficiency in ADH can lead to diabetes insipidus in dogs, cats, and horses. Additionally, an excess of blood in the circulatory system prompts the heart to release atrial natriuretic factor, which inhibits ADH release by the posterior pituitary, helping the kidneys excrete excess water. Next, we delve into the parathyroid gland and its role in regulating calcium levels in the body. This gland is essential for maintaining calcium homeostasis, which is vital for various bodily functions. The parathyroid gland consists of 2 pairs of glands located in the anterior cervical region. Chief cells, which are the primary secretory cells, and oxyphal cells, which are interspersed among the chief cells, play a significant role. Parathyroid hormone, PTH, elevates blood calcium levels, decreases blood phosphorus concentration, and increases urinary excretion of phosphorus. It also enhances tubular absorption of calcium, increases skeletal remodeling, and accelerates the formation of 1,25-dihydroxycholecalciferol by the kidney. PTH secretion responds to minor fluctuations in blood calcium levels. When blood calcium is raised, PTH production is reduced. Conversely, when blood calcium is lowered, there is a substantial increase in PTH secretion. Elevated phosphorus levels indirectly stimulate PTH secretion, and magnesium also plays a role in stimulating PTH secretion. The parathyroid hormone regulates calcium levels by acting on the kidneys and bones. It increases calcium reabsorption in the kidneys and promotes the dissolution of calcium phosphate crystals in bones, which raises blood calcium levels. This process is regulated by a negative feedback mechanism. Calcitonin is secreted from the parafollicular C cells of the thyroid. It is a polypeptide hormone composed of 32 amino acids, with its structure varying between species. The complete sequence of calcitonin is essential for its biological activity. Calcitonin helps develop varying degrees of hypocalcemia and hypophosphatemia. It primarily acts on bones and kidneys to lower calcium levels. Calcitonin's action is antagonistic to PTH on bone resorption and inhibits intestinal absorption of calcium. It functions as an emergency hormone to prevent hypercalcemia and protect against excessive loss of calcium and phosphorus from the maternal skeleton during pregnancy. Cholecalciferol, also known as vitamin D3, is synthesized from 7-dehydrocholesterol through skin irradiation. In the liver, it is converted to 25-hydroxycholecalciferol, and in the kidneys, it is further converted to 1,25-dihydroxycholecalciferol, the active form of vitamin D. Vitamin D increases the absorption of calcium and phosphorus from the intestine. A small amount of vitamin D is needed to allow PTH to exert its effects on calcium absorption. The primary target of 1,25-dihydroxycholecalciferol is the mucosa of the small intestine. Vitamin D-mediated absorption of calcium involves absorptive cells and goblet cells, which are essential for osteoclastic resorption and calcium mobilization from bones. It also stimulates the retention of calcium and phosphorus in the kidneys. Now, let's explore the pancreatic hormones, which play a vital role in regulating blood sugar levels and overall metabolism. These hormones are crucial for maintaining energy balance in the body. The pancreatic islets of Langerhans contain different cell types, each producing specific hormones. Alpha cells produce glucagon, beta cells produce insulin, delta cells produce somatostatin, and PP cells produce pancreatic polypeptide. Each of these hormones has distinct functions in regulating metabolism. In 1869, Paul Langerhans discovered the islets of Langerhans while studying the pancreas. He identified tissue clumps scattered throughout the pancreas, which he later suggested might produce secretions that play a regulatory role in digestion. Nearly all domestic animal species can suffer from diabetes mellitus, with insulin being a critical hormone for managing this condition. Insulin consists of two chains of amino acids linked by 3 disulfide bonds. Sanger was awarded the Nobel Prize for determining the chemical structure of insulin. Insulin is derived from a longer-chain polypeptide called proinsulin, which is converted to insulin in beta cells during synthesis and storage. This diagram illustrates the structure of proinsulin and insulin. Proinsulin is a precursor to insulin, consisting of A, B, and C chains. During the conversion process, the C chain is removed, resulting in the active insulin molecule composed of A and B chains linked by disulfide bonds. Insulin biosynthesis occurs in response to glucose stimulation in the ribosomes on the endoplasmic reticulum of beta cells. Glucose induces the translation of mRNA to produce proinsulin, which is then converted to insulin in the Golgi apparatus and packaged into membrane-limited storage granules. The biological activity of insulin depends on the whole molecule, with minor modifications of the disulfide linkage significantly reducing its activity. For glucose to enter the glycolytic cycle and be utilized, it must first enter the cell and be phosphorylated by ATP in the presence of hexokinase to form glucose 6-phosphate. Some tissues, such as nervous tissue and red blood cells, do not require insulin for glucose metabolism. Insulin is also not necessary for glucose transport across renal tubular or intestinal epithelial cells and the liver. Insulin stimulates the anabolism of carbohydrates, fats, and proteins. It decreases proteolysis, ketogenesis, gluconeogenesis, and lipolysis. Insulin carrier substances help transport glucose into cells, where intracellular machinery takes over. Insulin also promotes glycogen synthesis in the liver by suppressing the enzyme activity for glycogenolysis. Insulin promotes glucose transport into most cells in the body. It stimulates an increase in glucose carrier proteins, facilitating glucose entry into cells. Insulin also stimulates glycogenesis, the production of glycogen in skeletal muscle and the liver, while inhibiting glycogenolysis and gluconeogenesis, thus promoting glucose storage. Insulin increases the transport and conversion of glucose into fat stored in adipose tissue. It promotes the transport of fatty acids from the blood into adipose tissue and inhibits lipolysis, decreasing fatty acid output and promoting the storage of glucose and fatty acids. Insulin promotes the transport and incorporation of amino acids from the blood into muscles and other tissues, facilitating protein synthesis in cells. Additionally, insulin inhibits protein degradation. The release of insulin is regulated by negative feedback in response to blood sugar levels. Elevated blood glucose, such as after a meal, stimulates insulin release, while low blood glucose, like during fasting, inhibits its secretion. Glucagon is a protein hormone secreted by the alpha cells in the pancreas. It affects many of the same metabolic processes as insulin, but its actions are generally opposite. The liver is the major site of glucagon's action. Produced by alpha cells, glucagon is glycogenolytic and consists of 29 amino acids. It opposes insulin by increasing the activity of liver enzymes that activate phosphorylase, favoring glycogenolysis. Unlike epinephrine, glucagon does not affect muscle phosphorylase but acts on both hepatic tissue and muscle tissue. Glucagon release is triggered by hypoglycemia. Glucagon increases glucose production by the liver, raising blood glucose levels. It decreases glycogen synthesis, stimulates glycogenolysis, and promotes gluconeogenesis, converting amino acids into glucose. Glucagon stimulates lipolysis, breaking down triglycerides into fatty acids, and inhibits triglyceride synthesis, preventing fat storage in adipose tissue. Glucagon inhibits protein synthesis and stimulates protein breakdown in the liver. It has little effect on skeletal muscle protein but raises blood levels of glucose and fatty acids during fasting. Insulin deficiency, or diabetes mellitus, often occurs in dogs and less frequently in ruminants. Most avian species tolerate pancreatectomy except for owls, which suffer severely. In ducks, pancreatectomy lowers blood sugar significantly due to their high glucagon levels. Diabetes mellitus is characterized by hyperglycemia, glucose in the urine, and excessive urination. It leads to decreased carbohydrate utilization, increased fat and protein catabolism, weight loss, and other severe symptoms. Without insulin, glucose cannot enter cells, causing blood glucose levels to rise. This leads to glucose appearing in urine and increased urination, causing dehydration and intense thirst. Most body tissues cannot metabolize glucose without insulin. Nerve cells and red blood cells do not require insulin, but skeletal and cardiac muscles can only use some glucose without it. This leads to the body using fat and protein stores for energy. In diabetic animals, fat catabolism increases, leading to higher levels of ketone bodies in the blood and urine. This can cause liver failure and elevated liver enzymes. Ketone bodies, released as acids, must be buffered by the blood. They are excreted as salts, leading to a loss of sodium. Prolonged or severe conditions can cause acidosis, dehydration, and potentially coma. Protein catabolism occurs alongside fat catabolism, mobilizing body stores of protein for energy. This leads to weight loss despite increased appetite, as energy-yielding substances are depleted. Diabetes mellitus can be caused by genetic predisposition, hyperproduction of ACTH, abnormal nitrogen metabolism, chronic pancreatitis, or excess glucagon. Certain conditions in animals, like FMD recovery in cattle, can also lead to diabetes. PMSG is a glycoprotein with a higher content of carbohydrates and sialic acid. It has a half-life of more than a week and is secreted from endometrial cups. Fetal genotype affects PMSG serum levels, with horse fetuses having more and mule fetuses less. Endometrial cups form around day 40 and persist until day 85, increasing the development of follicles, leading to luteinization, accessory corpus luteum formation, and progesterone production.